It's not in presenter view yet.
Okay.
Yeah.
There you go. Looks great. Danica, whenever you want to get started.
Yeah. Welcome back from lunch, everyone. We have our final session this afternoon with another round of what looks to be great talks. And so we're going to get started with Renato Braghiere, talking about nitrogen limitation in the models. So go ahead. I will jump in again at 13 minutes and let you know.
Okay, thanks, Danica. Thanks for having me. My name is Renato Braghiere. I'm a postdoc at NASA Jet Propulsion Laboratory. And today I want to talk with you about how climate change impacts on mycorrhizae may amplify nitrogen limitation on plant growth globally. I want to thank all my collaborators that helped me with this research and DOE for providing funding. So just for us to start on the same page, what are mycorrhizae? So this term mycorrhizae comes from the Greek, myco meaning fungus and rhiza meaning root. So mycorrhizae is really this mutually beneficial relationship between a plant and root fungi. Basically, exactly like us, animals, fungi cannot convert electromagnetic energy into chemical energy, so they need plants for food. So basically what happens is that plants give out carbon through root exudates to these root fungi. And in return, these fungi go around the soil via hyphae, exploring and scavenging soil for water and nutrients beyond the root zone. So it's a win-win situation, really. So we as Earth system scientists, we think about this question quite a lot. So we want to know if the land surface will remain a carbon sink by the end of this century. And here on the left-hand side of this plot, I show you the results of 11 Earth system models in the CMIP5 simulations. This figure is quite famous. We've probably seen this figure a couple of times already during this meeting. But basically, this figure shows us that Earth system models disagree not only in the magnitude, but also sign of the land uptake by the end of this century. Another way of looking into this question is asking, okay, so if we get the change in land carbon predicted by these Earth system models, and we calculate how much nitrogen and phosphorus will be required to support that global productivity in the future, would that be sustainable or not? And according to this paper published by Will Wither and collaborators in Nature Geoscience, they found that net primary productivity does not meet the stoichiometric demands of plant growth. So including nitrogen and phosphorus would turn the land surface into a source of carbon by the end of the century. Well, in reality, we know that plants can access nutrients through different pathways, not only what is already available in there, but plants cannot move around. So they have developed a bunch of different strategies to acquire nutrients that are locked up in soils and getting all that nitrogen that is available in the atmosphere too. So the idea of the FUN model, and FUN here is standing for fixation and uptake of nutrients, started with nitrogen, but now we start thinking about the FUN model really moving towards phosphorus as well. But the main idea of the FUN model is that after we calculate the net primary production with our model, like CLM5, for example, we will use a part of this available carbon to acquire nutrients. So we spend a little bit of this carbon for nutrient acquisition. And the way in which we do that is by representing different uptake pathways. So in FUN, we can retranslocate nitrogen from leaves, we can fix nitrogen, and we can also have this mycorrhizae representation in there. So in FUN, we have the two most common mycorrhizae types, the arbuscular mycorrhizal association and the ectomycorrhizal association. And so we do that through cost functions. We calculate the cost, the carbon cost of acquisition for each uptake pathway. And we assume that the plant will always go for the cheapest one. Sort of, it's kind of like plants also want to save resources like us. And so the fun model was implemented in CLM a few years ago by Minji, Shi, and collaborators. And as you may know, when you implement a model that was built for a couple of sites into a global model, we run into a bunch of open questions. And one of the open questions that Minji and collaborators found, was how we represent the spatial distribution of mycorrhizal association globally. And so they come up with this smart idea where they associate either one type of mycorrhizal association or the other with blend functional types in CLM. So this table is rather like a binary association. So it's either one type of the other or the other, except for broadleaf deciduous temperature, where we have a 50-50 split. But when we plot this table into using the PFT spatial distribution for the globe, we can see this nice split in how the mycorrhizae is distributed around the world. So the extratropics are mostly associated with the ectomycorrhizae. And here in this axis, I show the ectomycorrhizae tree basal area in percentage. So every time you see red, it's only ectomycorrhizae. And every time you see blue, it means that it's the other type, arbuscular mycorrhizae. And so we have this this differentiation where the tropics are more arbuscular mycorrhizae associated and the extratropics ectomycorrhizae. Well, this has been validated with the global maps now, and in the past couple of years, a bunch of different publications use artificial intelligence and observation to recreate these global maps. But in the literature, we can see this split between tropics and extratropics for a long time. And by doing comparisons that I'm not going to show in here by sake of time, this PFT distribution, it represents quite well this spatial distribution. So now with fun in CLM, we can start asking lots of interesting questions about carbon costs and different nutrient acquisition strategies. So the first question I'm going to try to ask here is, has the carbon cost associated with global acquisition been changing with time? And to address that, we run CLM5 biogeochemical mode in the historical period with GSWP3 climate forcing, nitrogen position, and variable atmospheric CO2. We also perform some future runs using the SSP 585 scenario. The SSP5 is the pathway that we are currently on, assuming extreme fossil fuel usage. And we use CESM CMIP6 forcing. And so here in this plot, I show global NPP in petagrams of carbon per year from the beginning of the Industrial Revolution up until almost the end of the century, 2075. And the red line here represents the total NPP. So on the right-hand side of this plot, we see also a box plot. And the first box represents the historical distribution, and the second box, the future distribution. So we can really see how the CO2 fertilization effect makes NPP jumps from around 40 petagrams per year to 55. But the salmon line down below shows the amount of carbon used for nitrogen uptake. So we also see that this amount has been increasing with time, but the question is really, is the percentage of carbon used for an acquisition also increasing? And the black line tries to answer this question. So the black line here shows the salmon curve over the red curve. So it's basically, what is the percentage of NPP that is used for nitrogen acquisition? And it's in percentage, not in petagrams of carbon. So here we can see that around 1850, roughly a little less than 30% of all the NPP, the available carbon, was being used for nitrogen acquisition. Along the years, this value has been increasing, which roughly indicates that if we keep going on that direction, we will reach a point where 50% of the available carbon is probably being used for nitrogen acquisition. And so we start having a lot of open questions then. Will plants decide to shut down photosynthesis, for example, or they will start developing different allocation strategies to account for this extra usage on nitrogen acquisition? So that's something to explore there. Another question we may ask is, will ecosystems run out of nitrogen in the future? And to address that, we kind of use the same runs, but now looking at nitrogen demand and nitrogen uptake. And so there are two things to take out of this plots. First one is that nitrogen demand is always higher than nitrogen uptake globally. So global photosynthesis is limited by nitrogen uptake. But the second question, the second message is that nitrogen demand and nitrogen uptake are both increasing with time following the CO2 fertilization effect and climate change. So we came up with this risk of nitrogen limitation, which is basically we take the derivative of the historical curve for nitrogen uptake and nitrogen demand. And we basically say that if the rate of increase in nitrogen uptake is of the same order of magnitude than nitrogen demand, this area is not very much in a high risk of going nitrogen limited. Now, if the rate of demand is increasing at a higher pace than nitrogen uptake, then that area has a lot of potential to go to run out of nitrogen. And so we plot this figure here for each grid cell in CLM5, and we found that areas of savannas and forest grasslands transition zones present a higher risk of and limitation to plant growth, which is in alignment with values of nitrogen fixation peaking in those places. And finally, we want to know if climate change impacts, if climate change will impact the CO2 fertilization effect via spatial changes in mycorrhizal association. So to answer that question, we perform the same future runs using a present-day mycorrhizal map and a future projection of mycorrhizal map for 2070, taken from this study from Steininger and collaborators published in Nature a couple of years ago, where they found that in 2070, it is projected that the global will shift to a more arbuscular mycorrhizae. So basically, the amount of ectomycorrhizae association will decrease by 10% globally. But when we look at the boreal forest, we are talking about a reduction of 25% or more. So basically what it means is that while the globe is warming and those boreal regions are warming up, the arbuscular mycorrhizae will push the ectomycorrhizae species to the Arctic, where it's colder. And what is the impact of that for the global carbon cycle? So we plot here the delta NPP, so the difference between net primary production in the projection and the present day mycorrhizae. And we find that the boreal forest seems to lose part of this available carbon. And we have this huge decrease here in South American savanna. The Amazon, it's pretty uncertain. It looks like an open question. There are areas of the Amazon where we have increase, areas of decrease, but really we should pay attention to this area of the globe where the ectomycorrhizae is more present. When we sum up,
you're at 13 minutes, yeah.
Okay, so just to conclude, when we sum up, this 0.03 petagrams of carbon doesn't seem much globally, but we think this value is being underestimated for a number of things. So basically, nitrogen fixation in CLM follows GPP. So for the historical run, nitrogen fixation has more than double, and we assume that probably nitrogen fixation is alleviating nitrogen limitation. And and changes in flexible stoichiometry and P dynamics may also add on to the secondary effect of mycorrhizal changes. Sorry for going over time. I will take some questions. Thank you.
Thanks, Renato. We have time for probably one quick question. Looks like there's one in the chat from N King. It says someone presented the fun P model before. I'm wondering whether you have global predictions of P limitation and acquisition.
Yes, so we implemented the fun P model in E3SM, and we projected that yes, there is a decrease, but we haven't performed future runs with that, only historical runs. But we found in comparisons with iLab a better agreement for GPP, global GPP, and soil carbon. Yeah, but it will be interesting to perform future runs as well to see the impact of phosphorus on that.
Okay, it looks like Dave also has his hand raised if you have a quick question,
yeah.
Dave.
I was wondering with that last comment about the N fixation rising at the same rate as GPP, are you, is there some reason to think that that is an incorrect assumption, that it should be lower than that, or can you expand on that just a little bit?
So that's a pretty much open question, right? Like Will and Josh, and we've been discussing that offline, and it's kind of like, it makes sense that in the future, if nitrogen starts to get more expensive, the ecosystem would start fixing more nitrogen that is available in there. But we have a lack of observation. So a way to address that would be to have phase experiments, right? But phase experiments are quite limited in time and space. And also we have a few opposite results. For some sites, an increase in CO2 kind of shuts down nitrogen fixation, but for others, it increases in 300%. So it's pretty much an open question. Now, we've been thinking about trying to limit nitrogen fixation for present day values and do the same runs to see what would happen then. But we don't know. We don't know how nitrogen fixation will behave in the future. So it's pretty much an open question there.
Great, thank you, Renato. I think we need to move on. If anyone has additional questions for Renato, please enter them into the chat.